This is from space.com. Can humans reproduce in space? Mouse breakthroughs on ISS, a promising sign. The first ever study that shows mammals may be able to thrive in space. Keyword thrive there. So obviously there's humans who have spent basically a year or over a year in space on the International Space Station, mostly to kind of see the effects on the human body of being in space for that long, so that for missions such as going to the moon and the Artemis mission, but other missions such as going to Mars in later generations, kind of see how that could potentially impact the human body. And now with this beginning of mammalian studies about birthing and actual survival in space, we hope that this will yield good results. Now, this is from a Japanese team, and they had some interesting conclusions. We'll take a look at a lot of what they did in the actual experiments and how they were able to come to their conclusion. So, researchers have successfully grown mouse embryos aboard the International Space Station for the first time. It is the first ever study that shows mammals may be able to thrive in space. It is the world's first experiment that cultured early stage mammalian embryos under complete microgravity of ISS. In other words, this new mouse embryo study is a big deal because it suggests humans can reproduce in space someday. Once the first human baby is born off the surface of our planet, we would become a true spacefaring species. And like I said earlier, the landmark results comes as humanity prepares to return to the moon in conjunction with the Artemis program, which I've talked about previously. It's aims to land the first woman and person of color on the lunar surface by 2025, so just 2 years away. Building bases on the moon, doing prolonged missions on the moon, have people basically stationed on the moon, which would be pretty crazy. Um, Imagine having I say maybe a 10-day mission on the moon where somebody lands on the moon and then they get picked up and then they come back to Earth. Different than the Apollo program where you'll have the people actually uh going on the Earth, staying for on the moon, staying for a couple of days and then returning back to Earth. Whereas with the Artemis, they have actual bases that will be stationed on the moon, and possibly you could have week long or month long missions on the moon. Now, this rat study, or mouse study, I should say, does help us get a better understanding of how life can be impacted by external forces such as this. Now, we obviously have a lot of astronaut data on how it does to the actual human body, but this is a little bit different because these are mammal embryos that were hatched and cultivated in space. The experiment to grow the first mammalian embryos in space was led by Teruhiko Wakayama, a molecular biologist at the University's Yamanashi's Advanced Biotechnology Center, and a team from the Japan Aerospace Space Agency. The aim of the study was to determine whether a mammalian fetus can develop normally in the limited gravity or microgravity environment of space. So, a fetus, this is talking about a fetus. So, take this 5, 10, 15, 20 years down the line. Can humans be birthed in space? Can we, if, say, there is a mass global war or a mass pandemic or something of the sort, could we theoretically continue our civilization elsewhere, either on the moon, on Mars, wherever we need to go? Is it possible to actually not have life begin as a baby life on our planet. To find the answer, frozen mouse embryos were blasted to the ISS aboard a SpaceX Falcon 9 rocket in August 2021. After arriving at the space station, the early stage rodent embryos were thawed using a special instrument. Following this, astronauts cultured the embryos under microgravity for 4 days. The samples were then returned to Earth, where Wakayama and colleagues could study and compare them to mouse embryos grown in normal gravity here on terra firma, or Earth. And sure enough, according to a paper published in the journal iScience, I have that paper as well, I'll show you it later, the team reported that embryos cultured under microgravity conditions developed into blastocytes, which means a cluster of dividing cells made by a fertilized egg. So, good news with normal cell numbers. The research said in the paper that this clearly demonstrated that gravity had no significant effect on the blastocyte formation and initial differentiation of mammalian embryos. Now, interesting that they pointed out gravity in that particular quote right there, because gravity is not, when it comes to space and it comes to being outside of the Earth's atmosphere, gravity is maybe in the top 10, top 5 biggest concerns, but it is, in my view, certainly not number 1 in terms of concern. One of the major concerns for the early Apollo missions, and they were able to, fortunately, learn more about this by doing the Apollo missions. But one of the things that they ran into in the Apollo missions was a fear of being cooked alive once they got out of the magnetic field of the Earth. Obviously, gravity plays a huge role in the formation of, when we're talking about, in this particular study, the formation of mammals particularly, you need gravity, or at least we have evolved to require gravity in order to get our bones aligned, to get our DNA aligned, to get everything in our body aligned. We require gravity. Now, part of what this study has done is to kind of see whether or not that hypothesis is true. And it appears that there is some very good evidence to point to that maybe we could potentially see that there is really good evidence that potentially the beginning, or at least just the beginning, we're not talking about an entire lifespan, but the beginning of a lifespan, there is some evidence to point to that we could start a life in space and possibly take it for maybe a couple of months, maybe a year or so, on something such as the International Space Station. And then the next step is to take it to the moon. And then after that, take it to Mars. So very interesting stuff. According to a paper published in the journal iScience, the team reported that embryos cultured under microgravity conditions developed into blastocytes. This clearly demonstrated that gravity had no significant effect on the blastocyte formation and initial differentiation of mammalian embryos. The team also found that, if allowed, the blastocytes would grow into mouse fetuses and placentas while showing no significant DNA alterations or changes in gene expression. The survival rate of the embryos grown on the ISS was lower, however, than those cultivated here on Earth. So, interesting little end right there. Survival rate lower on the International Space Station. Now, with time, there is obviously a huge potential for different ways to get those survival numbers up. However, the beginning was, or I guess the foundation of what this study was trying to prove was is it even possible to have a fetus grow out in space? Yes, we want it to survive, but can basically a sperm and an egg meet up and then start to reproduce cell by cell into a much larger um, organism? And now this has established that at least for these mice, that is possible. Now, next step obviously is the survivability, maybe taking a mice from just the very initial stages, maybe up until birth, and then maybe from birth until however long, maybe till the end of its lifespan, which could be potentially years. So there is a lot to still do with this study and with this, I guess, avenue of research. In the future, it will be necessary to transplant the blastocytes that were cultured on ISS in ISSS in the International Space Station's microgravity into mice to see if mice can give birth. The team added in the paper, though saying this was a vital step toward confirming that blastocytes grown in microgravity are indeed normal. One aspect of the research the team will need to investigate further is the effect of radiation on space-based mammalian embryo growth, which is what I had talked about earlier. Though the researchers did look at this somewhat, they did not consider radiation exposure during the phases of live embryo cryopreservation and culturing specifically. All this knowledge may eventually become more important to humans when we finally begin to think about sending people to the moon and Mars for long ass stays. Now let's go look at the actual paper in itself. There's a couple pictures. Now we'll just kind of scroll through it. You can kind of see this little. They looked at these cells, see how they progressed through their lifespan on the ISSS, and you can see pretty much this is a graphical extract of kind of the mission. Embryo culture on the ISS. So they launched them at negative 95 degrees Celsius. They thawed. They brought them up onto the ISSS at 37 degrees Celsius. It started to I guess not even reproduce, but to start to or continue on its lifespan. And then they brought it back down to Earth, and then they studied the results of it. And that was pretty much what this study was in a very, very brief sense. But I'll put this down in the description so that you, if you want to, can take the time and read it. But what I wanted to kind of show right here is the timeline of this. So you can see 2021, the rocket launch is right here. So they're frozen at negative 95 degrees Celsius. Then they are thawed and cultured on the International Space Station about a week or so later. And then over the course of the time, they are then embryo fixation by PFA stored in refrigerated. So 4 days later, they are stored in a refrigerated. And then 20 days later is when they return to Earth. And then what is this? Basically 2 weeks after that, they are collected from ETCs. So this is kind of the lifespan of the mission. Yeah, you can see diagrams showing the timeline of this project. The dates are shown where the frozen embryos were prepared, launched to the ISSS, thawed and cultured on the ISSS, fixed with PFA, returned to Earth, and blastocytes analyzed. So, I mean, all in all, from launch, it took a little under 2 months for this full thing to go down. I have some more pictures that you can see of the actual little cells, the embryos themselves. And this is the embryos at the 2-cell stage before frozen. D is thawing by astronaut under microgravity. So I assume this is on that International Space Station. And then blastocytes collected from the ETC cultured on ground control. That's right here. Artificial gravity on the ISS and microgravity here. So you can see the difference between ground, artificial gravity. This is basically equal to the gravity here on Earth. And then this is microgravity. So basically the gravity that's on the International Space Station. So you can kind of see a little bit of a difference right now. Now, this goes back to kind of the survivability, right? Maybe once you get into those lower gravity fields, the survivability of these very small cells kind of goes down, but they're still able to, via this study, still able to survive somewhat. And here's the study. Once again, I said I will link this down in the description. Many other things through the study. They got videos, they got everything, but a lot of this is science that I do not understand. This is a video of them on the ISS unboxing it and actually thawing it out. But here's another study that I will link down in the description. This is another study talking about development of life beyond planet Earth. And this is kind of similar. This is basically the same exact thing. So potentially this mission, there is potential that if we are able to create different life in the vastness of space, say the moon missions, we land in 2025, and maybe by 2030 we're having full-time bases on the moon. Maybe by 2040, 2045, we're able to actually start having human babies, human fetuses grow and be birthed in space. Now, at the time, I'll be almost in my 50s when that happens, probably. If we're talking 2045, 2050, I'll be 52 pretty much in 2050. But there is definite real possibilities for this to continue and for them to use it in space. And I've talked about a couple of times on this podcast. Recently, I talked about, particularly if you remember the story that I did on the fetuses being grown, the artificial womb stories, if you can remember that. So maybe in the future, there can be some of that technology where you don't actually need the womb of a human to harvest a human fetus, but you may also not need it for animal fetuses as well. There was multiple different variants of the artificial wombs as well that I showed. I suggest you go watch that old clip. I have all the links in that video as well, so you can go look at the papers and look at the articles yourself and make your own determination. But I think that this is a small step, a beginning. Obviously, you want to get the survivability up. You want to make sure that these things are not dying instantaneously as soon as they get to space. You want to make sure that, yeah, you get some formation of cells, some advancement in the life cycle, maybe getting to a fully formed, in this case, mouse, but in the future, fully formed human, where they can actually be born and survive completely 100% in space. So, interesting start to the story, but we are not finished. Hopefully they figure that one out, because that will be really important to those Artemis missions.